Well, welcome everyone to our monthly AMS seminar. I am Jonathan Plett. I'm the president of the association, and it is my great pleasure this week to be introducing Dr. Meghann, who is going to be giving us our seminar. So she did her PhD at the University of Sydney and has now continued on as a postdoc. And she's been working within the mushroom industry since about 2016, looking at button mushrooms and oyster mushrooms and how to improve their growth. And specifically, she's interested in this interaction of fungi and bacteria and how that is affecting the nitrogen cycling within this quite interesting and controlled system. She's also been part of the AMS and our past fungal forays. If you go onto our website, you can see a lovely photo of her holding up, I think it's a stick from the Queensland fungal foray that she participated in. So it's really great that she can be presenting some of her other work here today. So with that, I will pass it over to you, Meghann, to talk to us about how you're making the industry more sustainable through your research.
Thank you very much, Jonathan. Good afternoon, everyone. My name is Meghann, and I'd like to take this opportunity to thank AMS for inviting me to speak today. So I'll be talking to you about bacterial-fungal interactions in Agaricus bisporus compost. And first off, I will just start with an acknowledgement of country. So we recognize and pay respects to the elders and communities, past, present, and emerging, of the lands that University of Sydney's campuses stand on. For thousands of years, they have shared and acknowledged and exchanged knowledge across innumerable generations for the benefit of all. So mushrooms come in a variety of colors, shapes, and sizes. As you can see in these photos here, some of these are wild edible mushrooms and some of these are cultivated mushrooms. But what they all have in common is that they are all delicious and they are all nutritious. But for today's talk, I will be mainly focusing on Agaricus bisporus, also known as the button mushroom. So how do we grow button mushrooms? So first we need some wheat straw, poultry manure, and gypsum. And we mix them all together and we wet it with some compost leachate and pile that onto a concrete slab. This part of the process is known as pre-wet and phase 1. It is a largely uncontrolled process, and it is mostly aerobic. I say mostly because there are still pockets of compost that are anaerobic in the profile. The compost is aerated either by manually turning or by supplying air through some spigots underneath the concrete slab. And our temperature profile starts off very mesophilic and then it becomes rapidly thermophilic, usually within about 24 to 48 hours. So after 9 to 21 days, the compost has been partially fermented. This then moves into temperature controlled tunnels, which is highly aerobic. So large volumes of air gets pushed through the compost profile. And there is no chance for any of these anaerobic pockets to show up. And it is highly controlled. At this point in time, it does go through 2 stages, so a pasteurization stage and a curing stage. And this whole process takes 6 days, and exactly 6 days. After that, our compost is then ready to be inoculated with mushroom spawn, which is the Agaricus mycelium that's been grown on millet or rye grain. And it takes about 16 days for the mushroom to completely colonize the compost. Again, this process is aerobic, as mushrooms are aerobic organisms, and they grow at an optimum temperature of about 25 to 30 degrees. Then the compost is then transported to mushroom farms or growing houses where they add a low nutrient casing layer known as made from peat moss and lime. And then after about 19 days, we have our first harvest of mushrooms, as you can see here. So I'm going to go into more detail about the temperature profile in composting. So in phase 1, we have a lot of microbes that are present from our raw material and also from the compost leachate. And these microbes are the ones that are responsible for rapidly generating that heat. And because they're using that readily available carbon and nitrogen that's there. The readily available nitrogen and also that heat releases a large amount of ammonia, which then gets used as part of the pasteurization process in phase 2, where our heat is then and the ammonia aid in killing any pathogenic microorganisms that are detrimental to humans or the mushrooms themselves, as well as any insect pests as well at that point in time. Then we move on to curing, where organisms that have survived the pasteurization process will utilize that free ammonia and incorporate it back into the compost as biomass. And at this point in time, we do consider the microbial community a climax community, as it is the established organisms there before the mushroom is inoculated into the compost. Then once the Agaricus has been inoculated into the compost, the mushroom is then using that microbial biomass as a key nutrient source, while also secreting lignin-degrading enzymes to break down our wheat straw. So what are the bugs that we get in the compost? So here we did a study in 2015 where we took multiple samples from multiple time points across the composting process, and we looked at the fungal and bacterial diversity throughout the composting process. So in phase 1, you see a rapid succession in fungal taxa as our carbon and nitrogen are used up. And then in phase 2 and 3, we get complete dominance by Mycothermus thermophilus before it gets taken over by the Agaricus midway through phase 3 composting. So Mycothermus thermophilus is a thermophilic ascomycete. It has been extensively studied since the 1990s for its growth promoting role with Agaricus. And it's mainly because it's one of the key organisms that use that free ammonia and does that incorporation during that curing step. It also produces cellulose and hemicellulose degrading enzymes. But this fungus is one of several bacteria and other microbial layers in composting. So with bacterial succession, again, similar rapid succession in dominant taxa in phase 1. And then in phase 2, we get a stable community working together to break down the polymeric compounds such as cellulose. I will be focusing mostly on phase 2 and 3 for this talk. So going into more detail about these organisms, Pseudoxanthomonas has been found in many cellulose degrading consortia literature, mostly because it produces beta-glucosidase, which does aid in breaking down cellulose. So it cleaves that beta-1,4 linkage in cellobiose and degrades that inhibitory compound that causes cellulose degradation to stop. It's not known to actually produce any other cellulose degrading enzymes at all. So the fact that it's there and it's there in quite a high concentration is quite interesting. Staurodobacter has also been found in a lot of lignocellulose degrading consortia in literature, and even some of the species from this genus are involved with denitrification. Last but not least, we have Mycothermus. This is actually a very novel genus that we discovered in our lab. It belongs to the Chitinofagaceae family. It has actually shown chitinolytic activity with Mycothermus, that dominant ascomycete. And it is also able to produce xylem degrading enzymes. So it does break down hemicellulose, which is important for our mushroom compost. So that was a very general view of the organisms during composting. But since we're having a little bit more of a closer look at phase 2 compost, So what I have here is the bacterial profile of 2 composting runs for phase 2, and they were sampled at the start of the phase 2 process, midway through the phase 2 process, just before the curing after pasteurization, and at the end of the phase 2 process. And the first thing we notice here is that Pseudoxanthomonas, again, is our dominant taxon, and it takes up to about 30% of the relative abundance. Most of this taxon is actually one particular species, Pseudoxanthomonas taiwanensis, which is a known heterotrophic nitrophile, which means that it produces nitrous oxide in aerobic and microaerobic environments, and can be a little bit of a concern considering that nitrous oxide is a greenhouse gas that is 300 times more potent than CO2. Another key organism is Chelatococcus, which is also a good cellulose degrading organism. It grows on cellobiose as its sole carbon source in a mineral medium. Its literature has also proposed that it aids in lignin breakdown. However, this hasn't really been confirmed yet, and is still being worked on at the moment. There is one species in this genus that is able to reduce nitrous oxide into molecular nitrogen. So that's good because we don't want that to accumulate. So we're hoping that we can use these bugs to make a more consistent composting process for our mushroom industry. But before I delve into that, I need to figure out if these organisms produce nitrous oxide. So I took some compost, isolated a lot of organisms, and sequenced them all and tried and found out what I had. We weren't able to get Staurodobacter. Mycothermus doesn't have any nitrogen functional genes in it, so we didn't include it in this. And I wasn't able to get Chelatococcus daeguanensis, but I did get Chelatococcus composti and Pseudoxanthomonas taiwanensis. So what I did was I grew these 2 organisms in a culture, collected the cells, and then inoculated them into a microcosm with phosphate buffer solution or compost, and added nitrite or ammonium as a nitrogen supplement into them. Because these organisms are predominantly during the curing stage of phase 2, I incubated these microcosms with those conditions, so at 45 degrees, with shaking to provide aeration. And then I sampled the headspace of the microcosm every 4 hours for 33 hours, and then analyzed for nitrous oxide in a gas chromatograph. And what we found was that when we gave Pseudoxanthomonas taiwanensis nitrite, it actually produces nitrous oxide. If we don't give it any nitrite, it actually doesn't really do anything, which was fantastic. And when we gave it ammonium, it also didn't produce any nitrous oxide, which is not shown here, but it's essentially the same as the control. When the Pseudoxanthomonas is added with the compost, we get a lot of our other heterotrophic compost organisms transforming that nitrous oxide that Pseudoxanthomonas taiwanensis makes into either molecular nitrogen or another nitrogen compound. So here in the black, we see phase 2 compost that has nitrite added into it, but without any extra taiwanensis, and it just straight drops, compared to our phase 2 compost that has the taiwanensis added to it, where we get a little bit of an uptick in nitrous oxide production, and then it goes all the way down. With phase 2 compost on its own, without any additional nitrite or any other additional bacteria added into it, we didn't see any nitrous oxide production. So that was excellent to see. Chelatococcus as well did not produce any nitrous oxide. So this is Chelatococcus composti in phosphate buffer solution. We're actually testing the Chelatococcus in compost soon. So with the ammonium, we do get a little bit of production made before, after 8 hours, it just drops and is essentially atmospheric. And similar to the nitrite, where again, we get a little bit of our nitrous oxide being made and then no production after 8 hours. Chelatococcus composti hasn't really been well studied. So a lot of its nitrogen functional genes are unexplored. We don't actually know if this species itself has nitrous oxide reductase in it. And from what we've seen here, it is also unlikely to be producing nitrous oxide. However, more exploratory confirmation needs to be done for that. So now that we've confirmed that neither of these bugs are producing nitrous oxide, can we use Pseudoxanthomonas taiwanensis and Chelatococcus composti to boost the Agaricus bisporus growth rate? So what we did, actually what a previous honours student in our lab did, was make race tubes. So she took some mushroom spawn, put it at the bottom of a test tube, and added an uninoculated phase 2 compost on top, treated with various things. So we had water, compost biomass, Pseudoxanthomonas only, Chelatococcus only, a mixture of all these, and including Mycothermus, which we know does boost the growth of Agaricus. She tried this with exponential bacteria and also stationary bacteria, and she measured the rate of growth every 3 days. So what she found was that exponential phase bacteria actually outcompete our Agaricus, and particularly Pseudoxanthomonas taiwanensis significantly affects our Agaricus bisporus growth rate. So that's number 3 in red here. It is significantly slower than our water only. Mycothermus, here we see, does give us our nice boost in growth rate compared to our control here. And it's actually most likely the main driver of that. Which is why we're seeing a very similar outcome with our inoculum mix here. But hang on, Agaricus is quite an aggressive mushroom or fungus. It's actually able to consume microbial biomass. Maybe it's because we're giving rapidly dividing cells that it can't really keep up. So what about if we give it stationary phase cells or resting cells? So again, that's what she did here. Our honours student grew our Pseudoxanthomonas and Chelatococcus to stationary phase, and then inoculated them at a rate of 5 × 10⁷ cells per gram of compost into the compost itself. And here we see Pseudoxanthomonas taiwanensis does boost the growth rate of Agaricus bisporus, and is shown to have the highest impact, although it's not very significantly different. But the significance, we might be able to see more significant growth or higher growth rate with this if we alter the inoculum concentration. So we suspected that our inoculum concentration was possibly too high, and we're basically stuffing the Agaricus mycelium with too much. So what we tried to do was take our inoculum mix, because that's what we wanted to do for the industry, and tested out a range of different inoculum concentrations. So here, this was our standard rate, which was 5 × 10⁷. And we found a significant increase in our Agaricus mycelium growth rate when we gave the when we inoculated the compost with either 50 cells, 500 cells, or 5 million cells per gram of compost. So if these 3 concentrations can improve our growth rate, does it also improve our mushroom yield? So moving on from our race tubes, we actually went on to do a growth trial. So took our best performing inoculum concentrations and inoculated that into 8 kilograms of mushroom compost and did our phase 3 spawn run at the Marsh Lawson Mushroom Research Unit at the University of Sydney. We also included water and also no other addition into our compost, which is our industry standard. So our compost was fully colonized after about 16 days, and then we added our casing on top. And what we found was that our highest inoculum concentration had a significant increase in mushroom yield across all of our harvest periods. So in our mushroom industry, one compost crop will be harvested 3 times. But the quality of our mushrooms at each harvest period varied, as well as we got a lot of patchy growth within the growing boxes of our experiment. And this patchiness may be due to either airflow issues in the unit or because the inoculum was added to an already high yielding compost. So it feels like we're not really benefiting it much because we're adding in these bacterial and fungal organisms when they're already there. But what about if they weren't there? Can we take say for example, a sick looking compost or a dysbiosis compost and add in our inoculum and see if that can improve the growth of our mushrooms or yield? Another thing was that we added our inoculum here after our climax community has already been established, where we might see an improvement maybe if we added our inoculum at the start of phase 2 and they go through that and establish themselves in the compost during this time point. So I feel the timing of the inoculum is also quite important as well for this experiment to work or to produce more significant results. So in conclusion, Pseudoxanthomonas and Chelatococcus does not produce nitrous oxide in vitro, which was fantastic to hear because we don't like making more greenhouse gas. Exponential phase bacteria compete with Agaricus for nutrient resources, and also our stationary phase bacteria actually boost the growth of Agaricus, which was good to see. Inoculum concentration is also important for optimizing our Agaricus bisporus growth rate, which may also be important for our yield as well. And research does what research does best. It continues to ask more questions. So what are some other key organisms in mushroom compost that correlate to an increase in mushroom yield? So currently we're working on trying to find a biomarker in our microbial community to see if we can correlate the abundance of one particular or several key bacteria or fungal organisms to an increase in yield. Inoculating a suboptimal compost or a compost that doesn't have this quite a nice bacterial profile at that phase 2 time point, and also the timing of our inoculum application, as well as looking at the nitrogen transformation and the functional genes in Chelatococcus composti. I'd like to thank Port Innovation and AMGA for funding our project, Kasia and Graham for their valuable knowledge and skill, as well as the composters who provided samples and advice for this work. Particularly Michael Curtis, my boss, who is the brains trust of this project, Phil, who was our technical officer with us, and especially Shiva, who did a lot of our race tube work to begin with. And thank you very much for listening, and I'm happy to take questions.
Great, thank you very much for that. Remember that you can pop your questions into the Q&A button at the bottom of your screen, and Meghann can take those questions as we come through. While we're waiting for those questions, I have one or have more than one, always do.
Yep.
So you were saying that the agaricus can eat the bacteria. So how does that work?
So it's kind of what they do is it sort of just it's not quite absorb. It doesn't quite phagocytose, but it kind of does that. I'm still trying to understand it. It's a little bit complicated too. The literature is really hard for me to wrap my head around on how it does that. But from my understanding is that as it grows, as the mycelium is growing, it's actually absorbing the bacteria and fungi that attach to it. Yeah.
Okay. So it's not killing them and then sucking them dry. It's
No,
literally.
I don't. I think the way that we see it is that the biomass is already, I wouldn't say it's dead, it's stationary.
Hmm,
And it's not so much, I don't think it's sucking them dry, but it could be.
cool. And then a follow-up to that, then, so you've done this addition of stationary phase bacteria of those three different genera. Have you tried just any other bacteria to see if any bacteria will do the same?
Yeah, we did try it with Bacillus subtilis, which is, our classic lab rat.
Yep.
And actually saw that it had the opposite effect. It actually caused the agaricus to be not happy, which was quite surprising considering that mushroom compost does have quite a few Bacillus taxa in it. But the Bacillus taxa are usually like Geobacillus or thermophilic bacilli.
Yeah, because if I remember right from your graphs before the agaricus is added, they're really going down at that point in
Yeah,
population, correct?
yeah.
Well, that's a good thing.
Yeah.
So you're also working with oyster mushrooms. Can you tell us a little bit about that?
Yeah, so we currently are working on looking at caffeine degradation, well, trying to circularize our waste streams. So we have the coffee industry produces a lot of spent coffee waste as well as silver skin, which is like the skin that comes off of the green bean as it's
Mhm.
roasted. And that itself is quite full of caffeine, quite full of nitrogen. And the coffee industry doesn't really know how to get rid of it. The way they actually get rid of it is just by burning it. And we're like, what a waste. So we grabbed some of their silver skin, mix it up, did a few different substrate mixes, and actually tried to grow some oyster mushroom on that. We're also looking at if the caffeine in that substrate degrades over time, do we get an increase in caffeine in our mushrooms as they're growing, which was quite interesting. Fun fact, yes, there is caffeine in oyster mushrooms when it's been grown on coffee waste.
Who needs
You
coffee
do have
before you
to
can have
eat,
mushrooms?
you do have to eat like 20 kilos to get the equivalent of 1 cup of coffee though. So
Oh,
yeah, you
okay.
sorry, can't make super caffeinated mushrooms. They're pretty cool though.
That'd be so
Um,
funny. People would be ordering their decaf and then having their mushrooms on the side and then not realizing they're actually getting caffeine.
yeah, exactly. Yeah, so we're doing that, and we're also looking at the microbial diversity as well. So what organisms are enriched when we give it this caffeine substrate.
Yeah, definitely. And if you could make oyster mushrooms grow better and be cheaper, happy days.
Yeah. So we do have one question. How many different varieties of Agaricus bisporus did you test the bacterial inoculation effects on? So we only actually tried it on one strain of Agaricus bisporus, which is our industry was our industry standard at the time, which was Agaricus A15. I think now they've moved on to a different strain. But yeah, it was only on one strain, and it was only on the white button mushroom.
So the industry really is reliant just on one strain at a time. They're not growing different types.
Um, Depends on who you ask, but no, they They mostly use one strain, uh, depending on which farm you go to or which compost yard you go to, but other compost yards will use a different strain.
Okay.
Yeah.
Yeah. And it's based, I guess, on the reliability and how much they're actually producing then is what how
Yeah,
they
and also the market. The market is a very key player in how many mushrooms are sold and how many mushrooms are grown.
Yep.
Yeah. These farmers are master manipulators on how to grow mushrooms.
That's fantastic. If only we could do that with so many other types of fungi.
It would be nice if we could grow some of our wilder mushrooms
Absolutely. I was
cultivated.
just talking with foragers this morning, and yes, they would love if they could have some of those wild grown mushrooms more readily.
Yeah, saffron milk caps. They are delicious. I
Absolutely.
do enjoy foraging. I don't get to do it very much, but I do enjoy foraging.
Very cool. Well, I think that's the end of our questions for today. Thank you very much, Megan, for your time, and thank you for presenting your work. We really look forward to seeing how it develops in the coming years.
Thank you very much. Thank you for inviting me.
Great. And thank you everyone for attending. Remember, again, we are here every month. We hope to see you again at the end of June. With that, have a great rest of your day. Thanks, everyone.